Well, we've mentioned the G protein coupled receptors, or GPCRs, or GPLRs, G protein linked receptors, the same thing, G protein coupled receptors. And the G protein coupled receptor family is a large family of genes which encode a large family of proteins, these G protein coupled receptors, and all of them have 7 transmembrane domains. And these are all alpha helices, which have hydrophobic stretches of amino acids in them so that they are integral membrane proteins embedded in the lipid hydrophobic bilayer. And there's an extracellular domain on the G protein coupled receptors, which have specificity based on their shape and their chemistry for signaling molecules or ligands. And depending on the tissue and the context of the G protein coupled receptor, the ligands can vary accordingly. So each G protein coupled receptor has specificity for a particular ligand. And we're looking at the G protein coupled receptor signal transduction pathway in broad terms, in general terms, at this point not looking at any specific ones. So how does the G protein coupled receptor pathway act? Well, when a ligand is bound to the extracellular domain of a G protein coupled receptor, that is when the cell that bears a G protein coupled receptor in its plasma membrane receives an extracellular signal of a particular type that is specific for a G protein coupled receptor, that G protein coupled receptor binds that ligand and changes its conformation in response. And that change in conformation causes a change in the G protein to which the G protein coupled receptor is linked and associated with. Now, the G proteins that we're talking about here are heterotrimeric G proteins. That is, trimeric means that there are 3 different subunits: the beta subunit, the gamma subunit, and the alpha subunit. And heterotrimeric means that although it's a trimer, each subunit of the trimer, each protein chain of the trimer is different. So it's hetero, not homotrimeric, but heterotrimeric. And these G proteins, heterotrimeric G proteins, are associated with particular G protein coupled receptors. Again, which G protein coupled receptor depends upon the cell in which we're talking about and the ligand that is specific for a G protein coupled receptor that we happen to be talking about. So what happens? A ligand binds to the extracellular domain of a G protein coupled receptor, and then, and then a conformational change ensues in the G protein coupled receptor such that this causes a change in the heterotrimeric G protein that we're talking about. Now, G proteins, as their name suggests, bind guanine dinucleotides or guanine trinucleotides, depending. So that's what we mean by G protein. So in its inactive state, the heterotrimeric G protein has GDP, or guanosine diphosphate, bound to the alpha subunit of the heterotrimeric G protein. In that state, it is inactive. But when it becomes activated by a conformational change in the G protein coupled receptor, which is caused in turn by binding of ligand in its intracellular domain, when this G protein becomes activated by a conformational change in the G protein coupled receptor, the alpha subunit of the G protein releases guanosine diphosphate and binds guanosine triphosphate. So it exchanges GDP for GTP. This is an actual exchange. We're not just phosphorylating GDP to GTP. Rather, we are exchanging GDP. We're releasing GDP, and GTP becomes bound to the alpha subunit of the heterotrimeric G protein. And in that state, that activates the G protein in a very particular way. It causes the dissociation of the beta and gamma subunits from the alpha subunit, which is now bound to GTP, guanosine triphosphate. And in that state, the free alpha subunit bound to GTP is capable of binding to and interacting with proteins called effector proteins. And it is these effector proteins that are responsible for transmitting the signal further into a cellular response. Now, there has to be, you don't want your signal, if once ligand goes away, you don't want to continue signaling to the effector protein through an activated alpha subunit of the G protein, because then the cell would keep responding even though there wasn't signal telling it to respond in a particular way. So the active state of the alpha subunit of the G protein is only transitory. And the alpha subunit actually has in it the ability, the catalytic activity to dephosphorylate GTP, to hydrolyze GTP back to GDP. So the alpha subunit of the G protein will hydrolyze GTP, releasing inorganic phosphate, and then what will remain is GDP. And in that state, the alpha subunit reassociates with the beta and gamma subunits, which were previously free, so as to form an inactive G protein. And that then allows this signal to be very transitory in terms of inducing a cellular response. If the ligand remains, that is, if the cell continues to receive signal, well then this process will be continued. But when the ligand stops, the alpha subunit is constantly hydrolyzing GTP and reassociating with the beta and gamma subunits. Now it turns out that the free beta and gamma subunits can also interact with effector proteins and cause other cellular responses, but it's beyond the scope of this class to go into that. We're going to focus on the alpha subunit bound to GTP and the effects it causes on effector proteins, such that the effector proteins will elicit various cellular responses. And that's what we're going to be concerned about right now. So what happens when the activated alpha subunit of a heterotrimeric G protein, that is, the alpha subunit which has disassociated from the beta and gamma subunits in response to binding of ligand to a G protein coupled receptor, what happens when that alpha subunit bound to GTP activates or inhibits, in some cases, effector proteins? We're going to look at two cases of effector proteins that become activated when an alpha subunit is in its active state bound to GTP and disassociated from beta and gamma. And we're going to see that these effector proteins that we're going to consider elicit a cellular response by the production of second messengers. And so we're going to look at at two examples of effector proteins and the second messengers that they cause to be released so as to elicit a cellular response. So that's what we're going to look at now. One of these effector proteins that can be activated by an active alpha subunit, it's an alpha subunit bound to GTP, is adenyl cyclase. And adenyl cyclase is an effector protein. Again, this is an effector protein, adenyl cyclase. And when adenyl cyclase becomes activated by an active alpha subunit of a heterotrimeric G protein, it causes the production of a second messenger molecule called cyclic AMP. And what adenyl cyclase in its active state does is take ATP and convert it to cyclic AMP. And we'll look at that in just a second. And then there are downstream effects in the cell caused by cyclic AMP. So second messengers are potent intracellular signaling molecules. Ligands are extracellular signaling molecules, but second messengers are intracellular signaling molecules. And cyclic AMP is a very potent signaling molecule inside the cell, a very potent second messenger. We'll come back and see what it does in terms of activating a protein here. So let's clear that. And now go to another effector protein. So here's another effector. Another effector protein, in this case called phospholipase C. And as you can probably figure out by its name, a lipase means an enzyme that will cleave lipids. And when phospholipase C is activated by an active alpha subunit of a heterotrimeric G protein bound to GTP, it cleaves a molecule called PIP2, or phosphoinositol diphosphate, into two second messengers. One, DAG, is called diacylglycerate, and diacylglycerol rather. And diacylglycerol, as you can see, is hydrophobic, and it remains associated with the plasma membrane because of hydrophobic fatty acid tails hanging off it. Just like PIP2 is anchored in the plasma membrane by its fatty acid tails. When PIP2 is cleaved by phospholipase C, however, the remaining DAG remains associated with the plasma membrane here. But the other product of cleavage of PIP2 by phospholipase C is inositol triphosphate. And IP3 is, as you can see, these yellow blobs here, these yellow circles represent phosphate groups. This is a polar molecule, and therefore it is freed to diffuse in the cell when it is cleaved from PIP2. And the IP3 second messenger diffuses in the cell, and there are receptor proteins in the endoplasmic reticulum that bind specifically to inositol triphosphate. And these receptor proteins are actually ion channels that open up in response to binding IP3. So IP3 comes down here, binds to these channel receptor proteins in the membrane of the endoplasmic reticulum. Now, the endoplasmic reticulum contains very high concentrations of calcium ions. And calcium ions are released when these channels open in response to IP3 binding. So when calcium is released into the cell, calcium is another regulator that can cause a cellular response in that it binds to calcium binding proteins. And when those calcium binding proteins are bound by calcium, that causes various cellular responses. So far we see that the activated alpha subunit of a G protein can interact with phospholipase C effector proteins, in this case causing the cleavage of PIP2 to diacylglycerol and inositol triphosphate, and inositol triphosphate can then cause the release of calcium ions through these membrane channels that open and allow calcium to flow out of the endoplasmic reticulum into the cytoplasm, where they can interact with or bind to calcium binding proteins and cause a cellular response. So thus far we've considered two effector proteins that can become activated by active subunits of the alpha subunits of heterotrimeric G proteins. One, adenyl cyclase, can cause the production of cyclic AMP as a second messenger, and phospholipase C, another effector molecule that can be activated by activated alpha subunits of heterotrimeric G proteins, causes the cleavage of PIP2 to diacylglycerol and inositol triphosphate, IP3 and DAG. So let's look at the molecules that are second messengers here. Let's look at cyclic AMP and its production by adenyl cyclase, and let's look at DAG and IP3 and its production by phospholipase C. Here is ATP, and activated adenyl cyclase, the effector protein, which is an enzyme, a membrane-bound enzyme, can cyclize ATP, releasing two inorganic phosphates. And what you see is, here's the inorganic phosphates here, that adenyl cyclase actually causes the linkage of this alpha phosphate here, this alpha phosphate, to the ribose sugar ring at the 3' carbon. So here we have an ester linkage to the 3' carbon, and we have an ester linkage to the 5' carbon here of what was ATP. But this molecule is called CAMP, or cyclic AMP, because it's cyclic in that the 5' carbon is actually joined to the 3' carbon through a phosphodiester linkage. So cyclic AMP is produced by adenyl cyclase when it is activated by an alpha subunit of a G protein that becomes activated by G protein coupled receptor binding to ligand. All right. Now, what about the phospholipase C enzyme that we mentioned? So phospholipase C here, this enzyme, which can be activated by an activated alpha subunit of a heterotrimeric G protein, converts PIP2 to DAG here, which is bound to the membrane. So this is PIP2, and you can see it's bound to the membrane by these fatty acid tails in the hydrophobic lipid bilayer. But when phospholipase C is activated, it cleaves off this part of the molecule, the phosphates here. It cleaves off right here, producing this molecule here, which is IP3, inositol triphosphate, and the other product, diacylglycerol, remains embedded in the plasma membrane. So we'll talk about what happens to this second messenger, cyclic AMP, and we'll talk about what happens to IP3 and DAG in just a bit more detail in the next part of this lecture.